Hello everybody at the KNBM lectures. Today's topic is: could a virus be a psychopathic killer, HIV, biology and pathogenesis. I want to invite you to look for the answer to this question. What HIV and Hannibal Lecter have in common? So let's start. Human immunodeficiency virus is part of Orthovirales order, member of Retroviridae family. Retroviruses got their name because they counter the Crick's central dogma. Reverse transcriptase is a unique enzyme enclosed within retroviral capsids and has ability to reverse the flow of genetic information. Retrovirus replication cycle displays several characteristics which distinguish this family from many other viruses. Integration of proviral DNA in the host genome is a typical example. Retroviruses may cause changes in cells, what is called transformation, and what can lead to oncogenesis. Human immunodeficiency virus, HIV, is classified according to its genetic structure and viral antigens into two types. There is HIV-1 and HIV-2. A large majority of HIV infections worldwide is caused by HIV-1, while prevalence of HIV-2 is restricted to some regions in West Africa. Both types can potentially cause acquired immunodeficiency syndrome. However, HIV-2 is less virulent and less infective than type 1. It is observed that time necessary to progress to AIDS is much longer in case of HIV-2. Both HIV-1 and HIV-2 are the result of multiple cross-species transmission of simian immunodeficiency virus, SIV, which infect African non-human primates. There's a schema of the phylogenetic tree of human and simian lentiviruses using the analysis of a pool region of lentiviruses. On the right side, in red, HIV-1 is displayed with its group M and O and P. The majority of transmission events was restricted to a relatively small group of infected people. However, in effect of SIV-CPZ transmission from chimpanzees to human, group M HIV-1 emerged and caused AIDS pandemic. 9 subtypes A to D, F to H, J, and K has been distinguished within group M HIV-1. They differ by at least 15%. But why? Recombination between different HIV subtypes and types is a common phenomenon. When one cell is infected by 2 virus particles classified to different types or subtypes, fragments of their nucleic acids may be exchanged. Progeny with mixed genome is referred to as recombinant. It has been estimated that about 20% of HIV-1 group M members are recombinants. They are called CRFs, circulating recombinant forms, and deposited in HIV sequence database. Intergroup recombination may also happen, for example, between group O and M in Cameroon. However, recombination between HIV-1 and HIV-2 has not been described so far. Okay, so structure of the HIV viral particle. HIV particles are round, about 100 nanometer diameter, and enveloped. In the bilayer membrane, key components of the HIV infection process are localized. The trimeric transmembrane protein, TM or GP41, anchors the trimeric of surface protein, SU or GP120. Together they form a knob-like structure, although binding between GP120 and GP41 is not covalent. Both glycoproteins are products of the ENV gene encoding a polyprotein. Below an envelope there is matrix consisting of the P17 matrix protein, Core capsid is localized inside matrix layer. It is built of the P24 capsid protein and contains two molecules of positive single-stranded RNA associated with nucleoproteins and enzymes bound to viral RNA. It's invertase and reverse transcriptase. There are 50 to 100 molecules of RT per virus particle. And next, it's genome. Retroviral genome consists of two identical copies, positive single-stranded RNA associated with NC, and it is localized within the core of the viral particle. RNA dimer explains why retroviruses are relatively resistant to UV and ionizing radiation, because every viral particle has two copies of all genes. HIV genome encodes three major proteins. It's ELM, GAG, and POL. Each of them encodes a polyprotein. After cleavages, fragments of them frequently perform similar functions. And there are several regulatory proteins. They are also encoded within HIV genome. Their expression is activated at different stages of HIV replication cycle. So maybe I'll give you a few examples. TAT, it's transactivator protein, promotes the expression of viral genes. VPR, which stands for virus protein R, is responsible for arrest of cell cycle, and VIF, viral infectivity factor, is responsible for successful infection in vivo. HIV genome is flanked by LTR, long terminal repeat sequences. Within 5' LTR is localized promoter for viral genes transcription that plays key role after converting into DNA and integration into host genome. Okay, so now we're gonna talk about the HIV replication cycle. So we have to start. Viral enveloped NOPE-like complexes composed of GP120 and GP41 are crucial for initial steps of infection. The HIV entry pathway starts with protein-protein interaction, GP120, and CD4 receptor. CD4 receptor, cluster of differentiation 4, plays a crucial role in communicating between T-lymphocytes and antigen-presenting cells. CD4 is a co-receptor of the class II major histocompatibility complex. A plurality of immune cells is CD4 positive. For example, T helpers, T cell precursor localized in thymus and bone marrow, dendritic cells, monocytes, macrophages, astrocytes. After initial GP120 and CD4 interaction, conformational changes occur that enable binding to additional proteins. The most common co-receptor of HIV are CXCR4, chemokine receptor 4, and CCR5, chemokine receptor 5, although more have been recognized. The first one is localized on the T lymphocytes and many other human cells, while CCR5 is present on dendritic cells, monocytes, macrophages, and activated T lymphocytes. The double binding provides stable attachment between the viral particle and the host cell surface. Interaction between GP120, CD4 receptor, and co-receptor triggers further conformational changes in viral GP120 and GP41 proteins. The second viral protein contains a fusogenic hydrophobic peptide on the N-terminus. In effect, cell membrane fuse with a viral envelope. Subsequently, the viral capsid is translocated into the host cell cytoplasm and partially uncoated. So, after that, viral nucleic acids and viral enzymes are released into the cytoplasm. Processing of viral genetic material can be divided into a few steps. So, firstly, reverse transcriptase converts positive single-stranded RNA genome into cDNA, complementary DNA. After that, RNase H makes endonucleolytic cleavages of the RNA strand in heteroduplex DNA RNA. And finally, DNA-dependent DNA polymerase activity of reverse transcriptase converts cDNA, single-stranded, into double-stranded proviral DNA. What is significant? DNA synthesis is slow and error-prone phenomenon. A mutation rate is estimated as at 1 to in 10 to 4 nucleotides per genome per replication cycle. Plenty of quasi-species are produced during a course of infection as a result. Because of substitutes in amino acid sequences, HIV properties are changing over time in the same host individual. HIV proviral dsDNA in form of PIC, pre-integration complex, is then transported into the cell nucleus, where integration into host genome takes place. Viral enzyme called integrase catalyzes this reaction. Place of integration is generally random. Provirus becomes a permanent part of host genome. There is no mechanism for precise excision of integrated proviral DNA. As a result, HIV infection is referred as persistent. During the HIV replication cycle, there is no classical viral RNA replication and no viral RNA replication. Proviral DNA directs the host transcription machinery to synthesize many copies of viral mRNA. TAT's function is to accelerate viral mRNA synthesis. This protein binds to the TAR, what stands for transactivation response element, at the 5' end of the HIV mRNA and stimulates further transcription. Viral mRNA is transported into cytoplasm, where it's translated into proteins and encapsulated into virus particles. Formation of viral particle is a stepwise phenomenon. Two positive single-stranded RNA molecules associate together with nucleoprotein, reverse transcriptase, and integrase. After that, structural proteins assemble over them. Immature virus particles migrate toward a host cell membrane and begin to bud, simultaneously acquiring fragments of host membrane enriched with GP120 and GP41. It takes about 24 hours from entry to release the first progeny viruses. Virus budding mechanism is quite different in case of monocytes and macrophages. They are accumulating within intracellular vacuoles and then released together into the extracellular fluid. Infected T-lymphocytes are eliminated in several ways. The first, exaggerated HIV viral particles production resulting in cell lysis. The second, activity of cytotoxic T-lymphocytes. And the last, because of cytotoxic HIV components in intracellular environment. Moreover, newly produced lymphocytes do not perform effectively their functions and to get targeted by circulating virus. Significant decline of Th cells is observed, so in effect symptoms of immunodeficiency are shown. HIV has the ability to lie dormant within infected cells. Latency is a state of convertible and non-productive infection. Expression of viral genes is stabilized on a relatively low level, so production of progeny particles is practically stopped. Infected cells with long lifespan, for example, memory T lymphocytes, macrophages, microglial cell, Cells seem to be important and stable latent HIV-1 reservoir. Production of virus particles by the cells may be dramatically accelerated as a result of virus activation. So now we're going to talk about AIDS pathogenesis and generally the course of the HIV infection. Prolonged HIV survival outside the bloodstream or lymph is unlikely. As a result, HIV transmission occurs in contact or transfer of blood, semen, free seminal or vaginal fluids, rectal fluids, and breast milk. Virus can also enter the body via mucous membranes, injured skin, and by parental inoculation. HIV is mainly replicating into infected cells, which transfer the virus regionally and to immune system cells, T cells, macrophages, dendritic cells, and so on. When transmitted by sexual contact, spreading into the bloodstream and regional lymphoid tissue is allowed by infected CD4 positive lymphocytes and dendritic cells. Few days after entry, virus can be detected in nearly lymphoid tissue. After about 2 weeks, it is detectable in the whole body, also in nervous system, and can be found in lymphocytes. After 3 to 6 weeks, non-specific infection symptoms are manifested, like fever, malaise, lymph nodes, enlargement, rash. This phase persists next few weeks and is followed by a symptomatic period. Immunodeficiency is usually observed when the CD4 positive lymphocytes decline to 300-400 per microliter. After a period of well-being, long-lasting illnesses become more frequent. Such progression to AIDS is a characteristic feature of HIV infection. AIDS is defined by complications such as serious infections or cancers. Mycobacterium tuberculosis, Toxoplasma gondii, herpes simplex virus, and cytomegalovirus are typical opportunistic pathogens occurring in AIDS. Neoplasm development such as Kaposi's sarcoma, carcinomas of the cervix and anus, B-cell lymphoma is also commonly observed. So, did you find anything what HIV and Hannibal Lecter have in common? Because I found few. The first is superficial charm because the structure variance is beautiful. The second is manipulative and pathological lying. I mean, providers, it's hiding inside a cell and cheating on immune system. The next is failure to adapt responsibilities for its own actions. Many T cells and macrophages are eliminated. Despite this fact, the virus keeps on infecting next ones. The next is lack of remorse or guilt, lack of empathy, and resisting any authority. HIV attacks also nervous system, and the last is just parasitic lifestyle. Thank you very much. I hope you get pleasure of this. So, see you later.